Hey, everybody. I would have written a much shorter intro if I knew they were going to read all of that. Thank you. Thanks, and I'm excited to be here to talk to you all about a lot about frogs today, and specifically about Rhonda foothill yellow-legged frogs. I think it's really neat. I'm getting to present with Sarah because long ago I read a paper that she wrote when I was an undergrad, and now I'm standing up here giving a talk with her on this very species, which is pretty awesome. So I feel really fortunate. I've had some really great mentors and colleagues, Amy Lind and Sarah Yarnell. People named Sarah or Amy generally have been the frog people I've worked with a lot. So we want to start with acknowledgements. We have multiple acknowledgement slides throughout this because we're sort of trading off with a bunch of different talk content. A lot of people have been involved in a lot of the work we're going to present. And so we're really thankful for all these folks for funding, field help, photos, etc. So just to give you an overview of our plan, we want to sort of trade off in about 20-minute chunks. And at the end of each chunk, we're going to have a little time for questions/stretch breaks, because 2 hours seems like a long time for us to just talk at you. And we want to make it sort of part of a broader discussion, particularly in the light that this species is somewhat of a hot topic for potential listing. So to encourage you to kind of talk about maybe what are you thinking about, why are you here, or what are you interested in, we're going to try to provide some conversation starters. So to start, we're going to do a little section on natural history. Breeding timing is really important. So how does flow influence reproduction? Ecology, just the general ecology and interactions of the species. Genetics, I'm going to talk about some of the new methods we're using to assess the species. And then population dynamics, which is kind of tying all of this together, like how does this all work? And then generally conservation outcomes. So that's the plan. So let's dive into natural history. This is just a broad overview. So most folks are aware we have over half the world's population lives within 20 kilometers of a permanent river. That means we have much higher use and impact on these freshwater systems. And as a result of that, we're seeing mean extinction rates are much higher in freshwater organisms compared to terrestrial. And there's a whole slew of impacts that we're seeing, but in particular, these main ones is from a paper by Dudgeon in 2006 that found overexploitation, water pollution, flow modification, species invasion, and habitat degradation were sort of the main factors, and they all interact with one another. And so how that relates to amphibians, well, amphibians, there's been a fair amount of literature lately talking about them, but in particular, they're really unique in that they link aquatic and terrestrial ecosystems. And from there's a great book I've been reading by Marty Crump on sort of myth and folklore of amphibians and reptiles, which is fantastic, and it talks about how we've sort of, amphibians represent a lot of things and a lot of cultures, but they are very much synonymous with sort of rebirth and creation and the bringing of rain, health, omens of good luck, omens of bad luck. But they are a really ever-present sort of symbol of this. And that's because they link two sort of different worlds, right? This terrestrial and this aquatic world. And they've persisted for a long time. It's one of the oldest taxa that we have currently around. And they persisted through what we consider the last 4 mass extinctions. And unfortunately, there's a lot of literature showing that we might be in the 5th greatest extinction right now, ongoing. And they're at the greatest risk. Of any taxa that we have, they're at the greatest risk of extinction globally. And there's a whole bunch of papers here, Vance Vredenberg and David Wake. Are we in the midst of this 6th mass extinction? This is from quite a few years ago now. Are we responding too late as we're detecting these changes? Are we detecting them sort of after we've sort of reached that point of no return? How we might be able to manage for these populations under climate change? On and on, right? There's a whole slew of literature that's coming out now that's painting a fairly grim picture of this really important and critical group. So let's talk about the species that we're going to talk about, which is Ranaboilii, foothill yellow-legged frogs. They are native to California and Oregon. This is the historical range. And what's really neat is they've been around, we think, for about 5 to 8 million years as a species. They're one of our only obligate river breeding frogs. So what's really neat is this species really needs moving water to breed in. A lot of amphibians are more lentic sort of pond breeders. This one, it definitively needs moving water all the time, but it can use it in a whole wide range of habitat, from really small, little tiny tributaries to really big rivers and everything in between. And unfortunately, they've disappeared from over 50% of their range, their former range. And this map here shows all those little yellow dots that you may or may not be able to see on the screen are the sort of current records. So if you look at the historical range in green, and then the yellow dots, that's where we have records of within the last 10 or 15 years of seeing frogs. And what you'll notice is there's far fewer dots in the southern part of the range, both on the coast and in the Sierras. And so we're seeing contractions on the south coast, the southern Sierras, they're mostly gone as far as we know. In Oregon, they've contracted. There are a few more records from there, but they've certainly contracted back in a lot of places. And in addition to that, at higher elevations, we're seeing contractions. So we don't see them very high up anymore. They used to go up to 4,000 or 5,000 feet, and now that only happens in like one place in the Sierras. And that might be because we've had really transformative change in California in particular, in two sort of major arcs that we'll talk about a lot more as we go through. One of the biggest ones was mining. The gold rush really transformed the landscape that we see today. And so we don't even actually have a good sense of what things looked like before the gold rush and the hydraulic mining that occurred. But assume that largely the Sierras are a very different landscape now than they were in, say, 1840. And then after that slew of change, we then, as more and more folks came here, and as part of mining, but also part of, we need water for farming, for agriculture, for drinking, et cetera, we started sort of plumbing our system with a whole bunch of different dams. And it turns out we're really good at engineering and building a system of water control and diversion. And so you can see that map is dams in California, and the pink squares are actual powerhouses, so generating power. So Amy Lind, as part of her dissertation, looked at these sort of specifically dams and range-wide changes in Boilii. And what she found in comparing sort of the modern versus historical locations of the frog, using landscape features and a bunch of attributes related to dams themselves, how high they were, how far they were from a certain spot, was that absent localities had far more dams upstream, and those dams were larger. So bigger dams and more of them, turns out we are seeing fewer frogs in that case. And then, and that was mainly in the Sierras where that trend sort of shows up. And in the southern coast, Andrea Adams just recently finished her dissertation, her PhD, and she has published a paper showing that she suggests that we think extirpation from the south coast may be because of a chytrid outbreak. So the fungus that you may have heard of that is largely fatal for most amphibians, frogs in particular, really sort of came through in a wave starting in about the 50s, which is similar to what we've seen in other species. And so she used a bunch of museum records and oral history from interviewing people. Really fascinating, like really dove into the history of things from sort of some unusual sources. It's a pretty neat project. But it does seem to sort of say that this chytrid outbreak did have some sort of impact on populations down there because they just don't exist anymore. So I'm going to go over the life cycle really quick because Sarah and I assume that everybody should know this, but we want to make sure that we are clear about how this species actually functions. So this is they're sexually dimorphic. So the little guy on top is the male and the female is below. They're in amplexus. So when amphibians mate, the female will basically, or the male will jump on the female and then she'll kind of hang out and find a spot to lay her eggs, and then it's external fertilization. And once the eggs are laid, this is one of my favorite photos, the one on the upper left, the little soccer balls. I love soccer. Those are eggs actually dividing. So that's an early, maybe like a few hours old egg mass that you can actually see cell division, which is pretty amazing. So now in this day and age, you can take a waterproof camera, get really close up and get a macro and take pictures like every few hours and actually see the eggs, the cells dividing, which is crazy. But anyway, eggs get laid usually in the spring. We'll talk a lot more about timing and where, but they're almost always attached to rocky substrates, from anything from like pebbles all the way up to bedrock, and occasionally some wood, if that's all they've got. And then they take anywhere from, 7 to 14, maybe even longer days to actually hatch out. Depends on water temperature and a number of other things. And then the tadpoles Sarah's going to talk a lot more about. They're super fascinating and really interesting, but they can take, a month to 2 months, depending on, again, temperature, food availability, a lot of other things, to metamorphose. And so you see the one, sort of the two guys on the bottom there are getting their legs. And once they metamorphose, juveniles take anywhere from 2 to 3 years, we think, to become reproductively mature. And so I do want to point out the upper left picture there, one of those frogs is not the species I'm talking about, and one is. So the one with the nice black eye stripe there is the Pseudacris, the chorus frog, and the one on the upper left is actually the Rana boylii. So that's a really nice comparison. Juveniles are about the size of a tree frog or a chorus frog, and when the chorus frog is full grown, right? So the adults can get much larger. So anyway, the juveniles kind of go off. We don't know a whole lot about where they go or what they do, but they sort of disappear for a couple years. Occasionally we'll see them back at the river the following summer. But then after a few years, then they come back as adults, and they are ready to sort of continue the life cycle. So what's really fascinating is recently we've realized that these frogs actually can live quite a while. So for a long time, it was sort of like, well, maybe 3-4 years, right? They make it out, spend a couple years, breed, and then they're done. Well, turns out some folks up on the feather have been using chin patterns to basically as a fingerprint, and they've found the same frog 12 years running. And we've seen similar incidences in captivity and a few other places as well. So these frogs actually can live quite a while in a really crazy dynamic system. Like rivers are, think about the winter in California, right? We could either, well, this year, maybe not so much, but occasionally we get some really big flows, some rain, the river comes up, and then sometimes not. But imagine living through that year after year. So pretty neat that they can spend this time, for this much time, potentially breeding every year, once per year, right? Okay, I'm going to pass it off to Sarah now.
So we're all fairly familiar with salmon migrating to spawn, that they leave the ocean and they swim upstream. Well, salmon are not the only ones migrating in and out of California's rivers. The adult frogs, though, are moving in the opposite direction from the salmon. So while the salmon are going upstream to try to get to tributaries to spawn, they're passing the frogs, sometimes the same time of year even. The frogs are hopping downstream from these dark tributaries that may be excellent habitat for adults, but are lousy habitat for tadpoles. And during the ecology section, I'll go into a lot more detail about why tadpoles need very wide, sunny, warm habitats in order to be successful. And we have some great evidence from people who have done radio telemetry on adult frogs. Ryan Bork and the folks at Garcia and Associates, and also Earl Gonslin have done studies where they put little radio transmitters on a beaded belt. And the longest migration that's been documented is a female who went from her little pool in a tributary to her breeding site, and it was over 7 kilometers. So not every frog moves that far, but routinely there are frogs that are moving 1,000, 2,000 meters between their little home in a trib and their breeding site. So the places where they go to breed are called leks. They all come together. Many males call, and these guys also call underwater. So it's not like chorus frogs where you hear this really loud croaking. Unless you have a hydrophone put underwater, you wouldn't hear them. And recently it's been discovered by a master's student from Chico, Courtney Silver, that they even call within the ultrasonic range in order for other frogs to hear when there's a lot of background noise of a waterfall or a very fast riffle. They have to call in a completely different sonic range in order to not be drowned out by the sounds of the water. And they go back to certain particular sites year after year, and those sites are often close to the tributaries where the adults live, and they have a very distinctive shape. They're asymmetrical, so one side of the channel might be steep, but the other side of the channel has a very gentle slope. And the reason for that is that it changes and it buffers the changes that happen when stream flow goes up or down. So in this cartoon, I've shown a frog, not drawn to scale, laying her clutch of eggs at two different flow discharges. So hydrologists use Q as the variable for discharge. So so this frog is laying her egg at Q1, and then at Q2, which is a lower flow, the water level drops, and so there's a change in stage, delta S. And you can see in these two shapes that for the same amount of change in discharge, you get a different amount of change in stage. So I like to use the analogy of two different kinds of champagne glasses. So, there's the traditional wide open champagne glass versus, the tulip-shaped flute glasses. And think about you either adding champagne to it or taking a sip out. When you take a sip out of the tulip one, for each sip, it's going to look like the height of the champagne goes down much faster. So if it's a river drying up and you have a U-shaped channel, then for that given amount of decrease in discharge, there's a much higher likelihood that the eggs are going to get exposed to the air. And it's the same thing in terms of buffering changes in velocity. So as discharge increases, then velocity is also going to go up, but it goes up at different rates based on the shape of the channel. And this suitable channel morphology is maintained by sediment transport. We all think of water flowing through a river, but the rocks are also flowing. And an example is at one of my field areas, there's a geomorphologist who did a tracer rock study. So he took rocks and he spray painted them orange. Each rock got a number epoxied in. And long after he left and stopped studying his rocks, I go back year after year to study the frogs, and I've been following his tracer rocks. And so, for example, one of his rocks, number 150, over the course of, say, the last 10 years, I have seen this rock literally move through 4 different breeding sites. So it shows up at one site, it sits there for a couple of winters, then we get a big winter, it moves to the next site. So the places that the frogs are choosing, they're both depositional habitats, right, because these rocks are landing there, but they're also active transport zones. So it's key to have this constant movement of rocks to maintain the channel shape. So when you contrast how a channel changes, if you have this natural disturbance regime where rocks are slowly moving downstream, you maintain these open cobble bars that the frogs like. But then when the flow is controlled by a dam, the disturbance is suppressed and bars get fossilized, they get overgrown by vegetation, you have large trees that grow up, and as a result, there's a loss of habitat. And in one of my sites, I've been following it for a very long time. I started during my dissertation in the early 90s, and I've got all of these different lek sites, and I know how many frogs are laying their eggs at each of these sites. And some sites just naturally have become overgrown by alders over the last 25 years. And so in yellow is the average of over 45 different lek sites, and the average over the last 25 years. There's about 15 or 20 frogs that lay their eggs at those sites. But then some of these other sites that started out being real hot spots in my study area, when they got overgrown and the canopy decreased, so it went from being mostly open, 77% open when I first started measuring canopy in the early 90s, to where it is now. And the trees have grown in, and for those sites, you can see there's a steady decline in those subpopulations. So now we're going to take a little break, and I'd like everybody to stand up and stretch and think about this question, talk to your neighbor about how this could be relevant for permitting projects like replacing bridges. So this happens to be downstream of a dam, and big trees have grown up to replace the bridge. A lot of trees had to be cut down, which was great. The frogs were like, yoo-hoo, we now have disturbance again. They went back and they bred there, but unfortunately, standard procedure is 3-to-1, 5-to-1 stem replacement ratio. So you can see these alder stakes in the foreground. So anyway, so take a little break, stand up, talk to your neighbor about what you would do.
So I'm going to talk about breeding timing and sort of the plasticity in that breeding timing. And mainly, how do frogs know when to breed? When do they spawn? And it's pretty fascinating. So environmental cues in these Mediterranean ecosystems. So in California, we have a Mediterranean sort of ecosystem where it's very dry in the summer, typically wet storms in the winter. And so you can imagine if you're a species or an organism living in that environment, you need to have cues that convey some sort of important information to trigger a response, whether that's salmon migrating or birds flying, frogs hopping around, whatever it might be. There are sort of things that they must be using in order to know that this is the time to do some part of their life history. And by the way, these photos that we're seeing are from a time-lapse camera that we have. This is from the North Yuba River. And so it's the same site in the same, basically over time. So this is August 2016. You're looking at the big picture. 2012 is down there on the bottom. And then this is, again, August 2012. And you can see it changed. So here's in the winter. And so, let me slow down here. So one of the other really important things about cues, environmental cues, they need to be reliable year after year. Think about California. We've gone from drought to wettest year on record, now back to probably drought, right? It seems like where we're headed. So that sort of constant change year after year, if you're an organism that's evolved in a system like this, you need to have a cue that's reliable despite those big changes. The other thing that's sort of really interesting about this is more and more we're seeing global climate change, although I put this on a slide with snow on it, is being implicated in shifts in these phenological or these sort of timing cues where we're seeing things migrate earlier, plants are emerging or blooming earlier, and those are all really related to this trend of it's getting warmer earlier, we're having shorter periods of cold, all the things that are being talked about right now. And so the question is, well, in a system like this, what triggers spawning in frogs, in river breeding frogs? And how plastic are those things? So how flexible are they interannually because of that variation I was talking about? So what's really neat about Rhonda is they're literally like little barometers in these rivers. They are really tightly tied, like Sarah was mentioning. Where they lay their eggs turns out to be really key and really tightly tied with the local hydrology and the regional changes in flow. And so that's because we've sort of, over much time and research, have figured out that they're really tightly tied to this cue of receding flows, sort of like the timing of when the flows start to drop, like Sarah was talking about. They're really picking that time because that indicates the future sort of low flow summer, which is great for tadpoles. When the flows aren't jumping around, that's when they want to lay their eggs. And so hitting that timing just right when the flows are dropping, not too fast, but they're dropping where they can lay their eggs and the tadpoles will enter the river right at the time where food is going to be available, turns out to be that's the cue that they're looking for. And so what is going to help them time that deposition? And what we've seen is that over 90% and probably more so, and same on the coast, of all the eggs we've observed tend to be in the same hydrologic habitat. So they are almost always in fairly shallow water, half meter or so or less, and very, very slight velocity, water velocity. They are rarely ever in an area where there is no velocity. Oftentimes when you take somebody out for the first time, they're looking in a place where there's like no flow. And that's not actually, they need some sort of flow to oxygenate the eggs. But it also needs to be in this sort of hydrologically stable place where if the flows do come up naturally or they do drop, the eggs will still be in a place that is not going to see that big stage change, right? So it's really amazing. You can go year after year. If you find a spot, find an egg mass, typically you can go back there and there will be an egg there almost every year unless something, the river changes, right? The sediment moves around. But it's really, when they find these spots, it's amazing how they can hone in on and know this is a good spot. And so the other question is, because this species has a huge range, all the way up into Oregon, on the coast, in the Sierras, those are very different climatic places. Are there phenotypes? Are there sort of a coastal breeding phenotype? Is there a Sierra breeding type? Some of the stuff I'm looking at with my dissertation right now, but I went ahead and pulled as many records as I could from a bunch of different reports and from all of Sarah's long-term data on the coast and Alameda and the eel, and then all the Sierra data that I could pull. So all the black dots you're looking at are Sierran populations. And basically each of those shows the date we first observed eggs in a given year. So years on the y-axis and the date is on the x-axis. And what's really fascinating is the Sierras tend to be about a month or more. The mean date of breeding is later, which makes sense. Snow melt, it's a much later sort of occurrence for the most part. Whereas on the coast, you have more of a rain-driven system, so they can go much earlier, right? We get maybe some spring rain, and then they're going to lay their eggs because in some of those places, they might dry up by the late summer. So there does seem to be a timing shift between these two things. But you can see at the very top there, the shift from 2011, which was one of the wettest years we've had, in the Sierras was out in July or late June, all the way over in 2014, and this extended even more in 2015, 2016, to the left where the frogs in the Sierras were breeding much, much earlier. So there's this massive shift from one year to the next and the timing of when they're laying their eggs. And so one of the things I asked was, well, what cues best explain that timing, that when they initiate oviposition? And how might river regulation, so when we put a dam in and regulate the flows, how might that impact these cues? And I'm not going to talk too much about the methods, but I used field surveys of egg masses. We did some modeling, looking at which variables were most strongly associated with timing. And then there's a really great tool now that's being more widely used called wavelet analysis. And using that as an index, so you can sort of combine that with some other things, this Colwell's metric, but it's a way to take a lot of time series data, and crunch it down into like a single metric or a couple metrics that you can compare across rivers. And it's a way to look at seasonality and predictability, which I'll talk a little bit more about. Okay, so this is from data from the American on the lower part there and the Yuba watersheds. We used paired watersheds, so we had some bypass reaches where they divert flow and the flows are fairly static for most of the year. A hydro-peaking reach where flows are up and down every day. So the stage change might be as much as a meter or more. And that's for generating power and for boating flows often. And then some unregulated rivers, so sort of what the natural flow regime currently looks like. And we basically took those and then collected metrics across all of those using logger data and gauge data to see what we could see. And so what I want to point out is what's really fascinating is the amount of variation that exists in a natural river. So look at that big pile of spaghetti. That is 70 plus years of river data, daily river data for the North Fork American, just up the road from here. And what's really neat is you see it's just a pile of spaghetti, right? But towards the end, in the spring, there seems to be a pattern year after year. Even though there might be a wet year or a dry year, you see that sort of long, curving, descending part of the hydrograph, right? And so if we highlight just the last 6 or 7 years, so 2011, one of the wettest years we had, you can see the snow melt is basically that part of that descending limb, right? That curve at the end was very late in the year. Versus the following year, 2012 doesn't show up very well, but as you go through this, you'll see the drought years are very low, very, very opposite of what you saw in 2011. And then as we keep going, you'll see maybe a slightly more normal year, but you see there's all this variation year after year, right? But every year, regardless, there is some sort of recession. And that's what the frogs seem to be using, because when we put breeding timing on this, we saw in 2015, some of the earliest breeding we've ever seen in the Sierras. And then in 2011, some of the latest breeding. Like I said, that's a 2-month span, which is pretty crazy that we saw that in the space of 4 or 5 years monitoring these rivers. And so when we plugged this data into a model, the things that popped out were the 7-day maximum mean water temperature, which was highly correlated with when eggs were laid, coefficient of variation, so how variable flows were daily, and regulation turns out to be important. We'll talk a lot more about that. And water year, obviously that was important because we have drought years and wet years, right? So if we look at this data, so all the little black dots in the background is just all the data thrown into this model, and the big dots are when we saw eggs, so that dot represents an egg mass. And almost all of the eggs, or not almost, all of the eggs we saw were above this 9 degrees Celsius. There seems to be a threshold. There might be some flexibility in that. For the most part, all the eggs we've seen are above that threshold. But then the flow tends to be, most eggs, like 90%, are below 10% coefficient of variation, or so they're in the less variable areas in the spring, which makes sense, right? The eggs are static. They're laid in a place that they don't want to have those flow fluctuations. So that's where we would see most of them. And that's because the river hydrology and regulation, the way this can affect things is what we're looking at now is a hydrograph of 3 different regulation types. So unregulated is in the blue. You can see these nice diurnal snowmelts, fluctuations early in the spring, and then that nice recession. A bypass, which is sort of just a flat line, right? And then hydropeaking, which is up and down every day. So there's a stage change every day. You can see it's quite big. And the reason that flow changes can affect these things, there are all these different pieces, right? Timing, frequency, magnitude. If you're a hydrologist, all these things have different metrics, and there's hundreds of these metrics that people calculate. But what's really important for the frog is if you were an egg mass laid in a river, and the flows drop, like Sarah was talking about, on the right there you'll see where just under that arrow is an egg mass. Later that day, the flows dropped. They dropped and totally dried out the egg. They desiccated the egg. So the little tiny pepper spots there on the bottom left are little tadpoles that have dried up. And so this is something we don't see very often, but you can imagine if you lose a clutch like this, one frog lays 1 egg mass per year, and that's it. And so you lose a clutch, and that entire clutch is gone. And the similar thing can happen, whereas if they lay their eggs at the low, and then the flows come up, it can actually scour, it can tear the egg mass off if the velocities come up too high. So the impact of regulation or these flow changes is very, very big. And that's why we're seeing the biggest changes or the biggest impact in these regulated rivers. So predictability and seasonality. I'm going to talk really quickly about seasonality is essentially think within a year. So think about in the summer we have low flows, in the winter we have high flows, but in a 12-month period we sort of have this seasonality, right? California's interesting, and I feel like we're down to like 2 seasons now, but generally there's some sort of seasonality. That's the pattern within 12 months. Predictability is the regularity of that seasonality. So how often every year do you have a winter storm? How often every year do you have that spring snowmelt recession? So using these two metrics, you can actually look at all these different rivers. You can compare them, and it's a really great way to do this. And so I used this analysis from this paper, Jonathan Tonkin. Great paper if you get a chance to take a look, but it's a wavelet-based analysis. We used all the flow data we could. So 10 plus years was the minimum of daily flow records. And what we did is took those same rivers that I mentioned earlier, so that regulated, the bypass, and the hydropeaking, and then we plotted sort of the predictability, remember the interannual, so like over many years, how predictable is the pattern, and the seasonality, so like what is that pattern within the year. And what's really fascinating is, if you look at the mean annual discharge over 75 years, it has a very predictable and seasonal pattern in the unregulated rivers. So you can see the two blue dots there in the upper right, North Fork American and North Fork Yuba, are two of our only unregulated rivers in the Northern Sierra. And if you look at a hydropeaking down on the lower left, you see it doesn't really have that same pattern, right? There's far less predictability and far less seasonality. It's just kind of a pile of spaghetti year after year because it changes based on demand, based on the year. And a bypass reach is sort of in between, right? So it might have a bit more seasonality, but it is not as predictable because they might have a spill or there's some sort of regulation that can still occur that is not predictable. So anyway, that's a really fascinating thing, and I'll talk more about how that relates to, I think, some of the genetics results we're seeing. But to summarize all this, what we're trying to say is flow turns out to be a really flexible spawning cue that's really important for this species. And that's because it can be applied in very different regions very effectively. Water temperature might be more of a threshold, and Sarah has a lot more data on tadpoles and temperature, which is also really a key component. And that I think what's really important to think about is a lot of these populations are struggling and are declining, and this might be because we're seeing mismatches from the flow, the regulated flows and or climate change, and what is actually happening in like a natural system. And we do see this where we see sort of the regulated pattern is very different than the unregulated. And so they might cue in on the wrong part of the hydrograph, right? Because it's at the wrong time of year, but the flow, temperature, and flow rate appears great. They lay their eggs and then the flows, come up or go down. So that's that section. So let's take a quick break, and I'd like you to think about if you're managing these rivers, which there are many purposes to manage, and we have, California is this really crazy, complicated place about how we manage our water, but how might we use some of this information to more responsibly manage a river system, balancing the need for hydropower and recreation and ag and all the other things that go into that? How might we do this in a way to incorporate or at least try to help out these critters? Leave it at that.
So I'll start now with the next section, ecology. And I'm going to be talking both about the OT ecology, so the interactions between frogs and their abiotic environment. And I'm also then going to be talking about the interspecific interactions that they have in their system. So mostly this is focused on tadpoles. The adults are eating insects, but the tadpoles are scrapers of algae. And you can see from the lower left photo, they have amazing mouths. So there's a beak that has keratinized parts that are for scraping and cutting. And then they have all of these rows of mini teeth. So each of those little rows of black lines, each of them, if you looked at it under a scanning electron microscope, would look like a little rake. So they're actually going along the rock surfaces and they're scraping up diatoms. And those are these where it says B, C, those are pictures of diatoms, which are little single cell algae that live in a glass house. They have what's called a frustule that's made out of silica. And diatoms are really different than plants in that they don't store their excess photosynthate as carbohydrate. So you think about a potato plant. The leaves are doing photosynthesis and the excess is stored as starch in the potato and we eat it. Well, diatoms, they store their excess production in the form of lipids or fats. So they're very oily and they have all these really great fatty acids that are very important for development. And then on top of that, there are some diatoms that are hosts to cyanobacterial endosymbionts. So little blue-green algae live inside the diatoms and they can fix the nitrogen that is dissolved in the water. So the food then is very high in protein because of the cyanobacteria, and it's very high in fat. So diatoms are the zone diet for tadpoles. And they prefer it. I've done cafeteria experiments where I offer tadpoles different kinds of food. They always prefer diatoms. And they're an important link then in algal-based food webs. So they take all of the diatoms that grow on the rocks or grow as epiphytes on larger algae, and they turn it into snakes. They're turning algae into dragonflies. They're turning algae into otters and birds and everything else that consumes tadpoles, because each of these egg masses can have thousands of eggs, and we're not seeing thousands and thousands of frogs. They're mostly getting eaten as tadpoles. And so they're very important in the conversion from algae to all the other things we like to see when we go on a river. So Ryan talked a lot about spring temperature, and now I'm going to contrast summer temperatures. We put, my colleague Alessandro Catanazzi and I put data loggers downstream of dams in a long longitudinal section, an equivalently long section of unregulated rivers. So are regulated. One bottom, we have a contrast. What you can see is as you gets warmer and warmer. All summer to reach regulated system. And this makes a big difference for the tadpoles. And so the lines on the base, that's showing when the eggs were laid, are in July and August. So to mimic what all of those different temperatures are, like close to a dam that's releasing cold water from the bottom of a reservoir, we raised tadpoles in different kinds of streams. So we chose streams on the Angelo Reserve, which is on the north coast in Mendocino County, and we forced the tadpoles to live in places where there's normally adult frogs, but they never lay their eggs there. So we chose these 4 creeks, and we raised the tadpoles in these flow-through enclosures. And in these dark shaded tributaries, the only way we could make the tadpoles grow and be happy is if we actually supplemented with algae from the sunny parts of the main stem. So we kind of just use them as incubators. If we tried raising them just on the algae that was growing in those dark streams, and they never made it to metamorphosis. So what we found was that there was a really clear difference in thermal performance. So in all of these charts, on the x-axis is temperature, and then on the top one, the y-axis, I know it's too small to read, but it's time to metamorphosis. So as the water temperature increases, so when it gets to be an average of 22 degrees C over the warmest 30-day periods, which is part of July and part of August, they could metamorphose in about 60 to 75 days. And in the colder streams where the average was 16 C, it took over 120 days to reach metamorphosis, and that's supplemented with high quality diets. So not only did they developed more quickly, they also grew to larger sizes. So the bottom two charts are showing their daily rate of gaining weight and then what their eventual size was at metamorphosis. So you can see that at the warmest temperatures, they're metamorphosing at about 2 grams. We also did common garden experiments. We collected embryos from multiple populations on the coast, the Sierra, regulated, unregulated rivers, and we reared them in growth chambers where we could manipulate the water temperature, both during the day and at night. And we also reared some outside in recirculating troughs. And what we found was that the inland phenotypes, not only do they have different phenotypes in breeding timing, they also are different in terms of their growth rates. So the Sierran ones, so those are on the left there, they all had higher growth rates. And obviously you can see at the warmer temperature, at 19.5 C, all the populations grew faster and put on more weight, but the Sierran populations actually had a stronger response. So they, the expression "make hay while the sun shines." Well, the Sierran tadpoles are really good at doing that. They respond very well to the increase in temperature. And so the graph on the left is just young tadpoles, and then for the ones that we raised all the way to metamorphosis in one common temperature, kind of a cool temperature, about a little less than 18 C, you can see there's a big difference in the body length increase from when they're recently hatching to metamorphosis. So the Sierran frogs are really different, not only in their breeding timing, but also in their capacity to grow as tadpoles. And what's even more interesting is we took all of these frogs that so we had ones that we grew at the Eel River in different streams. We had ones that we grew in the lab from different populations. And then we said, well, what temperature do they like? We made this funky linear gradient where we heated up, we took rain gutters and we put one end in ice coolers and the other end is actually sitting on a pancake griddle. And so because they're metal gutters and they conduct heat well, we got a linear gradient, cold at one end, warm at the other. And knowing where we saw the tadpoles in the gradient, we knew what the temperature was. So we had a quick read thermometer to make sure the gradient was linear, and then we just sat back and stood on a high stool and watched to see where the tadpoles were. And what we found was that tadpoles generally like it warm. That's what's on the y-axis here. T-cell is selected temperature. And then on the x-axis is the rearing temperature. And this is a compilation of hundreds of tadpoles that we tested. And basically what we found is that if they were reared at a really cool temperature, they compensated by choosing much warmer temperatures. So the ones that were reared at 16 C were choosing 22, 24 C. And then the ones that were reared at very warm temperatures reverted back to just sort of the baseline of liking it 19 or 20. So there are ways, not only in terms of the adults being plastic in their breeding timing, breeding location, but the tadpoles are also very plastic in their thermal preferences to compensate for conditions that are not optimal. So temperature has these direct effects, but temperature also has indirect effects because the algae and the diatoms are also responding to temperature. So there's an invasive species of diatom called Didymus. People also call it rock snot. And it proliferates where the temperatures are fairly cold and also where there's power peaking and the flows fluctuate. Didymus grows on the end of a stalk that creates lots and lots of mucus, which is why it's called rock snot. And this mucus actually helps it withstand the flow fluctuations. At night, when it gets exposed, it doesn't dry out because all the mucus helps it stay moist. So rock snot is proliferating in the Middle Fork American, and we did some growth experiments where we forced tadpoles to either eat Didymus that we collected, or we gave them, the superfood diatom, which is of the genus Epithemia. That's the one that has the blue-green endosymbionts. And then we also collected rocks from a river that has peaking flows, but the temperatures are warm. And there are native stalked mucousy diatoms, but much smaller than Didymosphenia. So Didymo is the one in the middle there. And I don't know if you can see the scale bar, but there is a scale bar there. And it's a much, much larger. The scale bar is over there on the left where it says 10 microns. So compare that 10 micron scale bar to their little scale bars in all the other ones. You can see Didymosphenia is huge as a diatom. And it has a very, very thick silica frustule, and it often just passes through the tadpoles. They eat it, they consume it, but they don't do well on it. And so we raise them at 2 different temperatures, and at cold temperatures, the tadpoles could maintain their weight on epithemia. So you see that's the 0 line, that means they just maintained. Anything above that line means they grew. So at cold temperatures, the tadpoles lost weight eating Didymo. If it was warm, they actually could maintain their weight on Didymo, but it's because Didymo is very unhappy when it's warm, and the cells start to die, and once they're dying and breaking open, then the tadpoles could digest them. So temperature affects their growth directly, it affects their food quality, but temperature also affects the tadpoles' susceptibility to being predators. So here we raise tadpoles at these ranges of temperatures, and then we expose them to nepids, water scorpions, which is, they're not scorpions, they're members of the hemipterans. And then we saw how did they survive, and what we found was that the controls and those exposed to predators, there was no difference if the temperature was warm. But at cold temperatures, they were much more likely to be eaten. And in part, it's because they're smaller and the predators can catch them better. But then there's also the effect of predators making it so tadpoles don't eat. Their main response when they're scared is they hide and they are very still. So there's a consequence to not eating. And the consequence to not eating is pretty dramatic at the cool temperature. So if you look at C, the graph all the way here to the right, so there's the 4 different temperatures at which we raised them. And you can see that at the colder temperatures, at the coldest temperatures, everybody got eaten. They're small, the predators gobble them up. But then as you go from 15 to 18.5 C, what happens there is that the growth is greatly affected in the presence of the predator. But then as it gets warmer, then the predators stop having a growth effect on them because they're able to grow and eat really well at those warmer temperatures, so the consequences of spending less time foraging are not as great. So there's a lot of really complicated interactions between predators and temperatures and food and trade-offs, so that there's sort of this zone where they can withstand predators if the temperature is warm enough. And then the next thing I'm going to talk about in terms of ecological interactions is disease. And this is getting back to the purported reason for why the frogs have disappeared in Southern California. So this is work I did with Andrea Adams in Alameda Creek. And we have Marcia to thank for spotting these sick frogs. And that's Marcia's photo there, actually in the lower left corner, of a very sick and dying frog. And what we found is that they were infected with the chytrid fungus, Batrachochytrium dendrobatidis, or BD for short. And it infects the skin, and you can see in the micrographs, there are these little round, I forget what they're called, sporocysts, and they release swimming spores into the water. So the spores have a flagella, and they swim through the water and infect other frogs. And in the Sierra, it's caused massive die-offs of mountain yellow-legged frogs. So in Alameda Creek, during the drought, we had a huge shift in where the frogs were hanging out. And this is what Alameda Creek looks like during flood and drought. The yellow circles are around the same sycamore tree with the Y-shaped or V-shaped trunks. So it goes from incredible flood to drawing into these small little pools. And this was the epicenter of where we were finding the dead and dying juveniles. So after the outbreak, we spent 2 years sampling all the different amphibians in the creek, and we found that they all had some level of BD infection. But it seemed like boilei was the most affected. And the prevalence of BD was higher where bullfrogs were present. So that's in chart A. And then it was highest actually where the bullfrogs were most established. In terms of water year, the prevalence increased as the drought proceeded. So from 2014 to 2015, the prevalence of infection increased. And we also found that males were much more infected than the other sexes or stages. And there was also spatial variation in the infection intensity. So the last slide was prevalence, what percent are infected. This is looking at BD load. So the colors in the diagram of all the different stream reaches that we looked at, the hot colors are the highest infection loads. So yellow, orange, and red, those are reaching the levels at which the infection can cause mortality. And you'll see the hottest colors are in the places where there's recent contact with bullfrogs. So the central population of the bullfrogs is in the reservoir, and then the bullfrogs sort of radiate out from the reservoir. And where the frogs are coming into contact close to the reservoir, either upstream or downstream of the reservoir, is where we were seeing the highest loads. And also loads were higher where there were more of the native frogs, which also makes sense. Think about, you put a lot of little kids in a preschool, the germs get spread very quickly. Well, it's the same sort of thing. So that's the thickness of the lines is just showing where there were lots of frogs based on egg mass surveys. So the thicker the line, the higher the intensity of infection. So we did modeling, and our mixed modeling showed that the biggest predictors of prevalence was the sex or stage of the frog, and that's, we think, because the males spend a lot of time at the breeding sites and they wrestle with each other. So they have a lot of skin-to-skin contact in addition to just being in a zoospore soup. Bullfrogs were also a very strong predictor of the prevalence and the water year type. So as the drought continued, so did the infection or the disease outbreak proceeded. In terms of load, one of the strongest predictors of how strong the infection was the mean daily stream flow. So when flows got low, that's when the loads got higher. Presence of bullfrogs also predicted load, and season. So when it was warm in the fall, the load also went up, and conspecific density was also a predictor. So the boilei are persisting now that the outbreak has ended, but the bullfrogs are still there in the system as predators, as competitors. They're also a vector and reservoir of the disease, and they also harbor other parasites as well. And here I've shown a big bullfrog tadpole with copepod parasites. And what's interesting is that when the bullfrogs had very high infection intensity, 96% of the boilei at that point in time had cleared themselves of the infection. So as long as the bullfrogs are in the system, they're going to keep the BD infection going. The yellow-legged frogs bask a lot and they clear themselves of the infections, but the bullfrogs are keeping it in the system. So I've given you sort of an overview of all the different kinds of ecological interactions between frogs and their consumers, tadpoles and their predators, and then with diseases. And so now I'd like to pose this thought question of how would you incorporate these kinds of ideas if you had to make a mitigation plan or a relocation plan, knowing sort of what their requirements are and susceptibilities are?
Well, it sounds like at least we're getting people to think about some things, which is good. So we're going to switch gears now and jump into conservation genetics, which is kind of the topic of my some of my research right now. See if I can get this to work. We're sort of switching slide presentations here. All right. Hopefully that's all coming through. Okay, so I'm going to talk about how to use, I'm using genomics, so next generation sequencing. You might hear a lot of buzz terms. I'll try not to throw too many of them out there. It's pretty amazing what you can do now, and I'm going to talk about some of those things. I think ecology is catching up to what genetics can tell us, which is pretty awesome. Another acknowledgement slide. That's my son. He's about 4 or 5 in that picture, and I think one of the reasons we do what we do is we both are fascinated by this critter and how complicated it is and interesting it is. But to me, I really want to sort of instill that in other people as well. And I highly encourage you, if you haven't been out in the field with kids, to do that. Go out there, show them what you do, or, they're just amazingly inquisitive, and it's really fun, and it sort of reinstills why I'm doing what I'm doing. I'm hopeful that, his kids might be, if he has kids, be able to see these frogs down the road too. So anyway, thanks to all the other folks. Mike Miller's my advisor, Sean O'Rourke, the Watershed Center, Brad Schaefer and Amy Lind, and all the folks who have helped out in the field. Okay, so this is the most text on any of my slides, I promise, and I'm going to read it for you because I think it's a really great quote by Mark Twain. "The face of the water and time became a wonderful book, a book that was a dead language to the uneducated passenger, but which told its mind to me without reserve. And it was not a book to be read once and thrown aside, for it had a new story to tell every day." And that's Mark Twain talking about the Mississippi. But I think the point is that, these systems are really multifaceted, and we've sort of touched on some of that already. And they really all have different stories to tell. And sometimes we need to sort of step back and understand and at least think about what that story is. So a very abridged history of Sierra Nevada rivers, which is the study I'm going to show you and the data that is from it. So rivers flow largely uninterrupted for 20,000 years, since the last glacial maximum, at which point, so about 20,000 years ago, most of the Sierras were under ice. From all the way down the southern tip of the Sierras all the way up to about the Feather watershed was under ice. Fast forward to 1853, when hydraulic mining started. Shortly after gold was discovered, they figured out how to hose hillsides down to extract all the gold from the gravel, and that was the quickest way to do it, was to set up these giant monitors, they call them, that they point at a hillside, hose down all the sediment, filter it out, and get the gold. It was banned not long after, in 1884, because there was so much sediment that was flowing down into the Central Valley and flooding a lot of the towns and the fields that were being used for growing food and other purposes, that folks really got upset and said, we need to stop doing this. So that was the next major change. Then, shortly after that, we started building dams and diversions, and through today, that continues to some degree. But we really started in the 1930s, and really about 1950s, really ramped up the infrastructure that we've built. Okay. So just to give an idea of what we're talking about, the scale, I know I've mentioned this hydraulic mining thing. Everybody's probably familiar with this, but I've been studying this, and it's still stunning to me to think about this. To give you a sense of the amount of sediment we're talking about that was moved, they based on calculations from mining records and other things, they think that over 8 times the amount of sediment that was excavated from the Panama Canal was basically washed out of the Sierra, just the northern part of the Sierras, just three watersheds, the Yuba, the American, and the Feather, 8 times more than was excavated. So 8 Panama Canals worth of sediment. That's how much we're talking about the landscape changed. And then we built a bunch of dams. And we're using those. There's a lot of large dams in California for hydropower. The expectation is with climate change and warming temperatures, residential energy demands are going to increase. We're already seeing that. So hydropower is actually becoming potentially a really important component of our energy system. And the problem is that we've shown a little bit of like what can happen when we regulate those flows. This is a hydrograph on the top over many years. It's just kind of that spaghetti pattern, but below it's zoomed in a bit to show you the flow pattern is really seasonal and very predictable, right? So I've kind of already talked about this a little bit. You see some winter storms, and then you see some snow melt that sort of pulses out in the early spring, and then that nice recession limb. Compare that to this. So this is from the Middle Fork American. I want to point out that this is before the dam was built. So this is the same river, the hydro-peaking river we've been talking about. That's what the hydrograph used to look like, actually. And there's gauge data to show that for many years, which is pretty cool. The dam was built in about 1964, I think, 65. So that's literally a year after the dam was built, and see how that hydrograph has changed. And we see that pattern of, up and down hydro-peaking. Just to extrapolate this point a bit more, I went ahead and pulled all the data I could from all these rivers that I'm going to show you before dams were built. So the USGS has an amazing set of gauge data dating back to the early 1900s in many cases. And so you can go pull gauge data for all these rivers before dams went in. And so this is that seasonality versus predictability graph that I showed earlier, right? So all the rivers are all way over on the high, very seasonal, right? They have the same pattern year after year. There's a sort of a gradient of predictability, but generally they have a seasonal pattern. Build a dam, and that all shifts. So this is now. This is from current data after the dam was built. And you can see the 4 dots or 5 dots over there, those are the only remaining, and actually that's just 2 sites. That's the same North Fork American and North Fork Yuba that are unregulated. Those are the only 2 that have maintained this signal. So the loss of this signal in many different rivers is really key to not just frogs, but fish and bugs and a lot of other things. Okay, so how does this all fit into genetics? Well, one of the things that we know is that these small populations have much more limited connectivity. If you have fewer individuals, they have a harder time finding other small groups of smaller individuals. And so that can have long-term effects on genetic health, right? And less gene flow, less genetic diversity, that can sort of bottleneck and cause all kinds of problems. And it reduces the ability for them to adapt in the future when you don't have as much genetic diversity to work with. So I've kind of covered this already. I'm going to skip through this. But I do want to reiterate that these make really good hydrologic indicators, right? So we know they're strongly linked with that hydrologic history, and that spawning timing and habitat selection are really tied to these cues. And so what I looked at is I took swabs of frogs, little mouth swabs, and we basically caught a bunch of tadpoles, and we took a little bit of their tail, basically the same amount like one of those water scorpions would nip off. That's about what we use. You really don't need much nowadays for this genetic analyses. And then I basically took that and ran some sequencing to look at all the markers within the DNA to sort of assess what is the overall genetic diversity, how related are these, lots of big questions like this. We use this method called RAD-seq or Rapture. I'll leave it at that. Ask me if you have questions. But this is a map of the rivers I'm going to show you. This is all from my dissertation research. I have sites like multiple tributaries within each reach and lots of different rivers. There's about 30 sites I'm going to show. And I just want to reiterate, here's that mean annual flow on the left, that bottom left, and then hourly flow on the bottom right. So you can sort of get a sense of the flow patterns. So the red is the hydro-peaking, the orange is bypass, and then the blue is the unregulated. And egg deposition matters because it basically affects whether they get scoured or desiccated. And so you can imagine if year after year these things get scoured or desiccated, the population sizes are going to decline, right? And they're going to have fewer frogs, so you would expect less diversity, smaller population sizes. So when you look at the genetics of that, this is across populations all the way, the Yuba, the bear, and the American. And what I want to point out, let me make sure to use this. What I want to point out is that the bottom left there, the pointer doesn't work, but the bottom left side, that's all the Yuba, right? There's 3 different watersheds all clustered together. All those dots represent a single frog. The top is actually the bear and the North Fork American, which are right next to each other, so you expect them to sort of cluster together. But what's really crazy is that really big purple group down on the bottom right, that's the Middle Fork American, that hydropeaking reach, right? So all the frogs from that are, even though they're immediately adjacent, and you would expect they should look like the Yubas there on the left, they're totally out on their own. So something's going on, and that dam's only been there, and the flows have only been in operation since 1965, right? So genetically, this frog could live 12 years, so that's not a lot of time to see a genetic impact. So that, I was pretty surprised to see that initially. Okay, so if we zoom in on the unimpaired, that North Fork American, you see this big scatter, right? And that's kind of what you would expect if frogs are moving around, they're sharing gene flow, and over the course of many years, even a frog from the very top of the watershed could potentially move all the way to the bottom, right? Tadpoles get moved around. That's what you'd expect. So you do see some structure in this. These are PCAs, but just look at the general pattern. So there is not a general structure pattern there. Look at the same set of data for the Middle Fork American, which includes data from the Rubicon, the Upper Middle Fork American, and this hydropeaking reach, and you see a much different picture. I think the main thing to look at here is on the left, all those dots are from the upper part of the watershed. Those are all actually from bypass individuals, and all the stuff on the right is from the hydropeaking reach. And actually, each of those clusters is from a unique tributary that's only maybe, 2 of those tributaries are only about a half kilometer apart. And we're seeing genetic differences between those populations. Not what you would expect. These frogs move around, can move around quite a bit, like Sarah said. They can move, 5-7 kilometers, no problem. Okay, so the hydropeaking sites are really divergent, which is striking. So another way we can look at this data is using this metric called FST, which is sort of a measure of isolation. And so what we can do is basically look at that across our different sites, and if it's 0, it means that they're all mixed up. They're completely the same, right? If it's 1, that means they're totally divergent. There's no similarities between them at all. And we're comparing, I just want to reiterate, we're actually comparing thousands, tens of thousands of markers, base pair little AGTCs. That's what the data goes into this. So it's really robust in that sense. And what I've done is plot this against just distance between any of these sites. So the mean distance from one site to all the other sites is what you're looking at. So on the bottom axis, it's just distance along the river. And on the y-axis, it's this FST metric, right, from 0 to 1. Just to give you a sense, most literature would say species are divided, FSTs greater than about 0.4 or 0.5. That's like different species, okay? That's flexible and depending on what method you're using. But just to give you a sense, that's about the threshold for different species. What you're seeing is a crazy pattern that correlates with the river regulation. So in the hydropeaking reach, we see the highest FSTs, even though those populations are separated in some cases by the least amount of distance. That's not what you expect. Theory would say that the further apart sites are, the bigger the FST should be. Okay, so that's pretty striking. And then one of the other ways we can look at it is we can actually measure genetic diversity and across these different flow types. And there's a couple ways to look at this. I'm showing each of these as its own population. So each of those dots represents a site, like a tributary or main stem site, wherever I went and sampled frogs. And on the x-axis, it's just a measure of diversity, the amount of genetic diversity. That dashed line is neutral. So what you would hope to see is, for the most part, populations are around that line. If they're to the left of the line, that means they're expanding, they're growing. There's additional genetic diversity being added to the population. If they're to the right of the line, it's bottlenecking. They're losing, so that's usually indicating a population's declining. almost all of the dots are to the right of that line. They're all bottlenecking. Even the unregulated sites, right? Like there are a few that are, and they're closer to that gray line, and there's one that's way out on the far left, which happens to be the main stem site. It's really healthy and pretty robust. But all the bypass samples, all the hydropeaking are way over to the right for the most part. So that to me tells me that most of the populations that I have, and this pattern applies to other, this is just the American bear and Yuba, but I've looked at the feather and a few others, and the pattern seems to hold. In the Sierras in particular, we're seeing this pattern of bottlenecking, where these populations are not doing well. And we might go out and see them, but the underlying sort of picture is pretty grim, okay? If we sort of summarize this in a box plot, so we basically took that same data and just squished it down by regulation type. Turns out they are significantly different across those different regulation types. And then I ran a model to then sort of reiterate to say, like, well, what is the best explanation for this really strong FST, this isolation? And what I found was that regulation type was actually the strongest factor, not distance, which is what you would expect genetic theory should say. And it actually was stronger than anything else. Like if you look at diversity, elevation, drainage area, sample number, stream order, I threw in a whole bunch of different things, right? Latitude, longitude, whatever you want to put in, the strongest predictor in every case turns out to be this regulation type. So to summarize that, I think the important thing is flow alterations really having this direct impact on this sensitive species at a genomic level in a very short period of time. And that the trajectory across the Sierras, not just in the unregulated, I think those sites are doing slightly better, but across the Sierras, they are not doing well. So that raises the question about listing, right? So I think flow management is important, and we've talked about, like, maybe can you mimic that recession limb and reduce those fluctuations, which is typically what's being written into most of the licenses right now. However, I think, that probably isn't enough. For a lot of these populations, in 50 years, if that trend continues, they're going to be gone. So I don't know. My question to you, and I just want to say this method is really powerful. It's great. I think there's a lot of things we can do to monitor populations over time. So if we do want to start doing translocations, Sarah will have some stuff about that later, that we can actually track and see, like, all right, are we being successful or not about sort of making this population healthy or bolstering it? Anyway, so the short answer is I don't know what the best answer is, but I do think there are some potential options we can play with. And the thing is, if we wait too long, I think we're already sort of getting to that point where if we wait too much longer, it's going to be too late for a lot of these places. And one other cool thing, I have 1 minute-ish, I want to point out, sorry. This is the Feather River, so similar, the same pattern on the North Fork Feather that's sort of the same flow pattern where it's totally outside, isolated from everything else in the Sierras. And then we actually just ran with Karen Goldberg and one of her grad students, Mallory. I was looking at Sierra yellow-legged frogs and Foothill yellow-legged frogs, because they actually, in the feather watershed, it's the only place in the Sierras where they coexist. So Bean Creek, for those of you that might have heard of this, it goes up to about 5,000 feet, and the frogs kind of hang out in the same river. And that one of the big management concerns is, like, are they hybridizing? What's going on? Because we think we have both species here, and they look the same. Like, if you had them in your hand, you can't really tell them apart. So we ran all the samples they sent us. And what's pretty crazy is we found 2 hybrids. But I want to point out that these are exactly 50-50. So these PCAs, if you were having actual gene flow, you would see it sort of spread out, right? Like but it's literally right down the middle, which means that those are essentially what's called an F1 hybrid, which is essentially like a dead end, right? Like, they may have succeeded that time, but they're not going to have successful offspring. So this is a really, really rare event, but we were able to pick it up. And I've done some additional analysis on this, and it looks like it's really, this is about 10 years of data. We picked it up, but it does seem to imply that occasionally you can have this sort of, and again, this is almost like a mismatch, right? Like, that's that clutch is no longer part of the population. So add that to the management question. So I will pose to you, knowing what you do, how might we manage, or would you recommend translocations? Would you recommend distinct populations? So focus on certain areas that are doing better, and maybe we stick with those, and the other ones that are really not doing well, we just say, well, there's not much we can do. What do you all think? So I'll take a couple minutes to answer that, and then we'll wrap up with like 10 minutes at the end to discuss.
While everybody's pondering distinct population segments or not, I'm going to try to pull it all together and talk a little bit about population dynamics and a little bit on a hopeful note about some opportunities for improving conservation, and then open it to discussion. So I'm going to actually not talk about a lot of the slides, but it'll be there, I guess, for the record. So basically, population dynamics simply means change through time. And just as we have incredible variation in hydrographs, shown here on the bottom, this is a hydrograph for Alameda Creek, populations also change dramatically. There can be periods with huge increases, and just in 1 or 2 years, you can have multiple, like 2-, 3-fold changes in population number from year to year, or 6-fold changes over a 5-year period. So given that range of variability, it is extremely difficult to use monitoring data to detect trends. And it also points up how powerful the work that Ryan is doing with genetics, because it is so hard. There's so much variability that in order to statistically show a trend, you need literally decades of data, and we don't have decades of time anymore to monitor these populations. So I've been monitoring this one population on the Angelo Reserve since the 1990s, and over the very long-term trend, you would actually, any kind of statistical model would show that there's no trend. But over the last decade, there has been a significant decline. And there's a 1-year autocorrelation. So how many frogs are laying eggs this year relates to how many there was the year before. But there's also a very significant 3-year pattern in the data that relates to what the survival was 3 years prior. So in the last decade, we had several very poor recruitment years. So in 2009 and 10, there were late rains, and more than 2/3 of the clutches were laid before the big storms. In 2012, we had really rapid recession, and 13% of the clutches stranded. The long-term average is 4%. Then we had 3 years where it was super hot in the summer, and the parasitic copepods went wild. And you can see some examples there of frogs that survived with copepods sticking out of them. But basically, they're the main drivers of the population appear to be the recruitment success. Brian and I compiled data that have, like my data, that have been collected all over the Sierra, and we just compiled all these data sets. And basically what we found is that in addition to the bottlenecking in terms of just the variability, even though we couldn't detect any kind of consistent trend just because it takes so many decades to do it, that there is much higher variability. So on the y-axis here is coefficient of variation, and we just compared all the regulated and all the unregulated. And it's pretty distinctive that the regulated rivers have much more variable populations. And it may be that by the time you could actually detect a trend, it's going to be too late, that they've already gone below some critical threshold. And so I'm going to skip through all of this part, but basically summarize all the threats that we've talked about so far, is that these small and fragmented populations are dealing with altered flows, altered temperatures, altered sediment transport, vegetation encroachment, channel incision. There's invasive species that are predators, competitors, parasites, disease, and we haven't even talked about marijuana cultivation and illegal flow diversions that affect them. So it does look pretty grim, but there are some hopeful signs and there are opportunities for restoration. This is Benbow Lake on the South Fork of the Eel River, and due to the budget crisis, it became apparent that the state parks could no longer afford to operate this seasonal dam, and it was cheaper to remove the dam than it was to repair it. So the lake has not been operated since 2007, and in that time, frogs have colonized these gravel bars. They're unvegetated, it's perfect habitat, and it's actually the most dense population on the South Fork eel. I thought I had a dense population on the Angelo, and they're like now 3 or 4 times as dense here. And then in 2016 and 2017, the dam was removed, and we collected egg masses and moved them around in kayaks to get them out of the construction zone, and now it looks beautiful. Another opportunity for recovery that's going on is in the North Fork of the Feather, where in the initial part of the relicense, there were these whitewater recreational flows that ended up scouring a lot of egg masses. And the population has been continuously monitored, and it unfortunately went from more than 30 egg masses a year in this 10, 12 kilometer reach to, in the last 2 years, 4 egg masses in 2016 and only 2 in 2017. So in addition to that lack of recovery, there's also bass and crayfish. And so in 2017, there was a pilot project. The last 2 egg masses were about to strand as the flows were dropping. So we put them into baskets, which you will recognize from all my previous experiments that were over. I took all my baskets from the eel. They moved to the feather after being sterilized and bleached. And the tadpoles were raised in them. And over the course of the summer, there are stepped releases, and that's what's in the chart there. So over 3,000 tadpoles that normally only 1% to 4% would survive if without intervention. 13% of the cohort was raised all the way to metamorphosis. So as crowding happened, we had to keep releasing and releasing, but it was a pilot, and it's going to be repeated this year on a much larger scale since it was quite successful. And there's also opportunities for education. The Marin Municipal Water District has done really excellent work in terms of fencing people and their dogs out of the creek. They've made great signs. They have a docent program. So on weekends, people stand next to the creek and teach hikers about yellow-legged frogs. So there are some hopeful signs of opportunities that we have for conservation. And now for the last 10, 15 minutes, we want to open it up to your questions and talk about the listing under the California Endangered Species Act and the federal ESA. Should we be focusing on distinct population segments? We haven't talked at all about forest practices, but these frogs do occur in places where there's logging. And then there's also reintroduction to sites where they're currently absent. One idea is to reintroduce them to Yosemite Valley. The National Park Service under Rob Grosso has done an amazing job removing bullfrogs from the Merced River and the other ponds in the valley. And so now it's kind of, they've started with red-legged frog reintroductions, and potentially yellow-legged frogs are next. And then, all of Southern California is the potential for reintroductions. So with that, I'd like to open it up and just say thank you very much to Ryan. It makes me so happy to share the podium with him. So questions? Things you want to... Oh yeah, okay. There you go.
Hi, I'm coming from the consulting field, and so a lot of our projects are going to be in areas with like bridge rehabilitations, size and retrofits, those kind of things, and clients being counties and cities and municipalities, and so this is like a big topic. right now it is being considered to be listed under CISA. And I know for the red-legged frog, there are accepted protocols and mitigation banks and this whole framework that's kind of established right now. Does anyone know where yellow-legged frog is in that? I know right now it's just a candidate, but have there been any established survey protocols that are accepted by the regulatory agencies? What are some steps that you guys would be recommending for clients out there who want to do the right thing? And I'll open that up to you.
Mike. So I can sort of answer that. I think it's a great question because there have been a number of sort of protocols written. I know PG&E had Craig Sultanrich and Alicia Poole had a protocol that was sort of for all amphibians surveyed in rivers. They got used for quite a bit for relicensing. Sarah Yarnell and I and Amy Lind have written a protocol for surveys that's on the Center for Watershed Sciences website that has both the data sheet and sort of a written protocol about basically if you're surveying for egg masses or different life stages, what's kind of the minimum data that you would want to collect that includes some of this geomorphology and habitat types. So I think that's a great place to start, but I totally agree there should be some kind of standard, which is kind of we wanted it to be some of the standards. But I do think it depends on the study too. Like what's the question you're after? Is it just to sort of establish presence or absence? Is it to sort of track long-term trends? Is it to sample for looking at the genetic health of the population? I think there's kind of some minimum data that could apply to all of those things, but then there's also additional pieces that might be important. So, yeah, I think the why the red-legged frog has a kind of a nice established thing is it has a federal listing behind it, which helps, right? So there's some standards that had to go into place, and so potentially I do think this is a good thing in the sense that we are going to hopefully be forced to put some of those things into place, regardless of the listing outcome. Yeah, do you have any?
Well, the other thing I wanted to say is that sometimes they are extremely difficult to detect. And I actually am very hopeful that collecting environmental DNA could be incorporated into protocols because, actually, in Earl Gonsolin's Masters, he did this really great comparison between visual encounters and detections of radio-tagged frogs. So he would compare how often he saw them to, how often he okay, they're here somewhere under a rock. And more like 50 or 60% of the time when he went out on days that he thought would be good detection days, so, it's warm enough, it's sunny, all of the things that would bring them up, they're under the rocks 60% of the time. And you can't turn over every single rock at a site. So the reason egg masses are so great is because they're really obvious. Sometimes. I mean, yeah, sometimes you have to snorkel, and sometimes, you only know they're there because you stick your way up under a big boulder and you feel the squish of the jelly. But they can be extremely cryptic and difficult to detect. So I think if and when a standard protocol is made, there's going to have to be like a number of times that you go out with no detections, or even including some sort of eDNA.
And I do want to mention, so Karen Goldberg up at Washington that I'm working with, they actually have eDNA data from all the feather where the hybrids were. So they're actually using eDNA to sort of as also a double check. So we use sort of genomic data. They're taking DNA from the water to determine which species are which and where they are. And so that's been successful for foothill yellow-legged frogs. So going forward, and I know the National Park Yosemite is also using some eDNA as well to sort of see if they can find it in some of these historic locations. So I definitely think that'd be a really great, if the idea is to just see are they there, I think that in combination with actual, egg mass surveys, like Sarah said, is really you kind of need multiple options. And for egg mass surveys, I would almost always recommend snorkeling because they are often in places that you can't see or even feel without being in the water. Yeah, oh yeah, please.
The other really amazing thing about these populations is that they're able to persist at extremely small population sizes for a fair amount of time. So on the Tuolumne River, downstream of Hetch Hetchy, there's been a lot of effort to sample for them over the last 10 years. And so, 1 year, Ryan found one frog. Another year, then, 5 years went by. I found 2 tadpoles on one day. And then just this last spring, one of SFPUC's biologists found one frog in one tributary. So over 10 years, there's been literally 3 or 4 sightings, and the rim fire has happened. All of these things have happened, and somehow they are hanging on by a thread, but they're there, but extremely difficult to detect. So I don't know if that is a good or a bad thing for your clients, but it's really, I think actually if they were there historically, I would say, they still could be there. In the microphone.
Yeah.
So we have read your protocol, and also we were just wondering if you have a project, what is the distance from the project? We kind of, Grant and I are reading your protocol like a mile. Is that the distance? And is that a mile downstream, a mile upstream, a mile total from, say, a bridge replacement, or?
Great question. I would say, so for instance, I've done a bunch of the sort of FERC relicensing, and the nexus to the project is, like 200 meters or 300 meters from like the main stem, right? So we'd only survey 300 meters or whatever it was. More and more, having done my research where I actually crawl up those tributaries for like a mile, I find way more frogs past that confluence, that little stretch, right? Like unless you time it just right, like Sarah was saying, and you see they're breeding there,
Right.
the odds of finding something are pretty low in just a 300-meter stretch. I think so. It's sort of coupled between, like, again, what's the goal? Like, do you want to just establish that they're there? Do you want to see if the habitat could support them? I think, yeah, like a mile is a fair, a kilometer is actually a fair chunk of at least to sort of say, like, roughly are they in this kilometer is a good amount. And the other thing is, repeat visits becomes really important because you could be out there and it might look great, but the odds that you miss them is surprisingly high in a lot of those places. Unfortunately, and I also recognize like crawling a kilometer up a tributary turns out not to be super easy a lot of times. So you're kind of limited on certain things, which is, again, eDNA could become sort of another, and there's not 100%, it's not a fail-safe thing, but as a way to supplement a visit, I think it becomes really important.
And by the way, I was the crew leader on Mallory's project, and this is like the first time I've seen results, so that was kind of cool.
Yeah, if I can just continue on this one. So determining presence is a big factor, but also avoidance and minimization measures.
Right.
And so I think that was one of the questions that were proposed up there. If you guys could kind of talk to any measures that we could incorporate into our projects.
Well, I think it's really important to think about timing. So if you know that you're going to be mucking around in July, you might actually have to do your minimization in April when the frogs would be coming to breed. So a lot of it, I think, is that it's much harder to do, once you have thousands and thousands of tadpoles or lots and lots of little metamorphs, it's, egg masses are easy to move out of harm's way.
Can I just add on that real quick,
yeah.
though? So because of its listing right now or proposed candidacy, we can't relocate them, is my understanding, without an incidental take permit, which can be costly. Is there a way that we can get around that?
Well, Laura, I'll defer to Laura.
Like 6 to 9 months to get.
Right.
It takes about 6 to 9 months to get.
Well, yeah, I don't agree with people to ask about the regulatory side, but yeah, I mean, the reality is that's probably, up having to do, but yeah.
Okay.
I think moving eggs is kind of the ideal
But
option.
sometimes it's not possible.
Yeah,
Like we
totally.
had to use 2 different strategies at Bembo because the flows were pretty high. in 2017, and our whole method with the kayaks wasn't working. And so then we ended up having to use block nets and catching tadpoles and moving them. And it was much more labor intensive and much more difficult.
One more question, and it looks like it's 3 o'clock.
Anybody?
All right, thank you, everybody.
Thank you.
Right.